In this first exercise of the prac, we're going to be looking at stomatal conductance from our two plants here. We have our well-watered dwarf bean plant and our water-stressed dwarf bean plant. We're going to be looking at water loss along that leaf-atmosphere boundary. To do this, we're going to exercise the leaf from each plant and then look at the change in weight over a 5-minute period of each of those leaves. To cut off the leaves, we're going to use a sterilised razor blade and a chopping board. Once we chop off these leaves, we're going to immediately weigh them and then measure the leaf area using something called the leaf area meter. Don't get your leaves mixed up as well. So I've just weighed each of our leaves, and now we're going to measure the surface area of each of these leaves. This is very important to do because obviously a much bigger leaf is going to lose a lot more water than a much smaller leaf. To measure the leaf area, we use the leaf area meter here. I'm going to grab one of our leaves and place it on this backlit screen here. I can then grab a sheet of glass or a sheet of Perspex and place that on top of the leaf, making sure that our leaf is well spread out over that area and not folded back over itself. We also have a ruler on the screen here, and this is just to help calibrate our instrument. If we change the height of this camera here that films the leaf, we will need to recalibrate the instrument. Over on the screen here, we can see a picture of our leaf on that backlit screen. If I press grab here, that will take a photo of that leaf. On the left-hand side of this screen here, we have a few different boxes, and these give us options to click on the leaf, and that will select the area that we want to measure. This first area I've selected has all turned blue, but we have not yet covered all of our leaf surface area, so let's keep clicking away at non-blue parts of our leaf. We need to continue doing this until we're happy that the blue area represents the area of our leaf. There we go. So once we're there, we can press this measure button here, and that's going to spit out the area for us of our leaf. Very importantly, make sure you also record the units on this as well. The units might change depending on how it's calibrated. In this case here, we can see it says millimeters squared. In this prac, one of the things we'll be comparing amongst our light plants and our shade plants is the stomatal density. This is the count of stomata on the underside of the leaf per leaf surface area. The way that we can do this is by taking a leaf peel. To do this, we want to tear the leaf, and as we're doing it, we want to sort of try and break it away on an angle, like I've done here. It's a bit messy, this one, but this transparent or translucent bit of leaf we can see here is the epidermis, and that has all of the stomata on it. We'll put this onto a microscope slide and add a drop of water to it before putting our cover slip on top. We can then put that slide under a microscope and start counting the stomata on the epidermis there. So we can start off looking at the stomata under the 4x magnification. We can zoom in further to get a clearer image, but be mindful as we zoom in more, our surface area on the leaf is going to change. At 10x magnification, we can start to see the stomata quite clearly. These are characterized by those two kidney-shaped guard cells around the outside of the stomata. On some microscopes, you might see a bit of chlorophyll in these cells as well. At 40x magnification, we can see those cells or the stomata really, really clearly, and this is the magnification that would be ideal for counting the stomatal density. When you are counting the stomatal density at 40x magnification, make sure you do look at a few different fields of view for each leaf slide that you look at, and this is just to account for variability within each leaf or each slide that you make. One of the things we'll be comparing amongst our plants that have been grown in shaded conditions against those that have been grown in light conditions is the chlorophyll content. In this analysis, we're going to get a known area of leaf from each plant, and we're then going to grind that up and mechanically break it down. We'll dissolve those contents in some acetone and then place this into a centrifuge so we can remove all of those solid parts. We'll then put the liquid component into a spectrophotometer and we'll get some absorbance readings at 2 different wavelengths. Based on these absorbance readings, we can then calculate the chlorophyll content in each of our leaf samples. So with the chlorophyll analysis, it's really important to use a particular size of leaf. We want to know the size of leaf that we're using because a bigger leaf is obviously going to have more chlorophyll than a smaller leaf. It needs to be quite a particular size though, so that when a bit of leaf goes into our spectrophotometer, we have an amount of chlorophyll that's within the range that the spectrophotometer can read. We can use a cutter like this to cut a standardized area of leaf. We can simply push it down onto our leaf, give it a good shake like that, or run it around and round, and we'll therefore get a nice circular cutout from our leaf. We can place that bit of leaf onto our leaf area meter. Here's one that we've cut out just previously. And then we can use our leaf area meter to measure that. We've selected our areas of the leaf that we want to measure. When that's all turned blue, we're happy that that blue area represents the piece of leaf that we've cut out well. And over here, we've pressed measure, and we can see the area of leaf there. In this case, we have 0.2796 centimetres squared there. So I have my piece of leaf area here, which I've cut out. It's a bit less than 2 centimetres squared, and it's important that it's this size because if we use a leaf area bigger than this, there will be so much chlorophyll in the sample that it will maximise the detection threshold on the spectrophotometer. I've also measured this under the leaf area meter so I know exactly what size this is so that I can accurately quantify how much chlorophyll I have per leaf area. The first thing I'm going to do is mechanically break this leaf down. To do this, I'm going to mix it with some acetone and a pinch of sand and start grinding. So I'm going to add 10 mils of chilled acetone here. And I'm going to add a pinch of sand in there as well, which will help break it down as I grind it. Put that down and start grinding away, and after a few minutes you should have your leaf completely broken down. So after a few minutes, you should notice that your sample starts to turn a green colour, and that's already indicative of the chlorophyll content in the leaf. We can then decant this into a centrifuge tube and place this into a centrifuge. This will separate all of the solid components, such as that sand we used to help break down the leaf, down to the bottom of the tube. We can then take the top or the liquid component and put this into a spectrophotometer and read some absorbance values of this sample. So I now have my sample into a centrifuge tube. I'm going to place this into the centrifuge so that it spins with an RPM of 5,000 and it's going to go in there for 5 minutes. Make sure you label your tube as well before it goes in so it doesn't get mixed up with the other tubes that will go in at the same time. Importantly, make sure you balance the centrifuge as well. So now that we have our centrifuge sample, we're going to put that into a glass cuvette. It's very important we use a glass cuvette. If we use a plastic one, the acetone will start to dissolve the plastic. We can use a plastic pipette to transfer some from the top. Be careful trying not to transfer any of the solids that we've just worked to get down to the bottom. So we might need to do a few goes. Before our leaf sample goes into the spectrophotometer, we need to blank the spectrophotometer. We're going to use a cuvette filled with acetone, since our leaf sample is dissolved in acetone, to blank the spectrophotometer. We can just press the 0 button on the spectro, which will do that for us. Now that we've zeroed the spectrophotometer, we can place our leaf sample in there to get some absorbance readings. I'm getting 2 absorbance readings on the spectrophotometer here, one at 647 nanometers and one at 664 nanometers. If you look at your lab manual, based off these absorbance readings, we can calculate the chlorophyll content per area of leaf that we've used in our sample.